That's sort of the background and the principles, concepts. It went very fast, but if you come to the Salvador course, you can, I have more discussions on those. So one of the things that you have to do, another point that I make in my Berkeley course and Salvador course, is when you do a molecular epidemiologic study, you have to ask a question first. Before you begin a study, ask a question. Don't just start typing organisms and then say, what am I going to do with these data? Which is very frequent, in the US, here, everywhere. That's what people do. They just start typing the organisms, and then you say, what do I do with these data? And I say, what was your question? Okay? So ask a question first before you begin any molecular epidemiology or epidemiology study. Ask a question. So here we ask a question, talking about drug-resistant urinary tract infection. What is the source of drug-resistant E. coli that causes community-acquired urinary tract infection? And is drug-resistant UTI associated with clinical use of antibiotics? Very simple questions. Okay? So UTI is a big problem, all over the world. In the U.S., about 7 million office visits a year, more than 100,000 hospitalizations per year. There's about $1.6 billion in expenditure, medical expenditure. It's the most common bacterial infectious disease of women. How many of the women in this room has had UTI? See? This happens every time. Whether I ask this question in Berkeley or Japan or Brazil, no woman raises their hands. And I don't believe it. Okay? And at the end of my lecture, then the women say they're more comfortable and they raise their hands. So you don't have to be embarrassed about UTI. And I'll tell you why soon. So, okay. So we did a study in Berkeley done by a student named Amy Mangus, who wanted to actually do a study of sexually transmitted infections. And I said, I don't know anything about STIs. I don't want to work on STIs. I used to work with E. coli diarrheas, so we compromised. So let's work on E. coli UTI. Okay? All these diseases occur in the same anatomical area of the body, right? So UTI was a good topic. So we found 505 urine samples and isolated 255 E. coli isolates from 228 women. Okay? And first we looked at the drug resistance, and tranethyl sulfa resistance was 22%. And this was the drug that was being used empirically in that clinic at that time. But with 22%, that's above the cutoff recommended by Infectious Disease Society of America to switch to some other drug. So now in this clinic they use ciprofloxacin. Okay? So this is not that interesting. But then we looked at the genotype of those tranethyl sulfa resistant E. coli isolates. Okay? And we used a simple method called ERIC2 PCR, which we'll talk about tomorrow. Okay? It's a very quick way to screen hundreds of E. coli. Okay? And so we found, for instance, these two strains here, as you can see. Oops, I'm still not used to this. Okay? The identical pattern by this method. Oops. Okay. And then we obtained comparison isolates from other parts of the U.S. Okay, from University of Minnesota, University of Michigan, to collect their isolates to compare to the Berkeley isolates. And we did a PCR, again, ERIC PCR, and just put them on the same gel. So you can see that many of them are completely identical. Okay? There are thousands of these patterns, and yet from these three different campuses, which are about 2,000 miles apart, we found similar or almost identical patterns. So Amy thought, initially Amy believed that UTI was a sexually transmitted infection. So when she found all the similar isolates in Berkeley, she said, well, maybe there's some Berkeley male student is infecting all these women. Okay? But then when she found that the same isolates were found in Michigan and Minnesota, 2,000 miles away, then she started having doubts. Okay? How could a male student from Berkeley go to all these other campuses? Impossible. Or maybe in the United States, maybe not impossible, but highly unlikely. And so we began to suspect, okay, what's going on? How did this clone of E. coli spread to all these campuses? Maybe it was a technique. Maybe ERIC PCR is not a very discriminating test. So we used PFGE. And even by PFGE, most of the ones from California almost had identical, closely related to other isolates from other campuses. Okay? By Tenderverse criteria, which I'll talk about tomorrow, they're all closely related. And so from Berkeley, 51% of the trimethylene sulfa isolates had this pattern we call CGA. Okay? So if this wasn't for this clone, instead of the prevalence of resistance to this drug being 22%, it would have been 11%. Okay? Single clone was responsible for doubling the prevalence of trimethylene sulfa resistance. Okay? And similar rate, 38% in Michigan, 39% in Minnesota. And they belong to different serotypes. They all belong to ST69 by MLST. Okay, we'll talk about that tomorrow. They had these so-called virulence-associated genes. They were multi-drug resistant to ampicillins, streptomycin, sulfonamide, tetracycline, and trimethylene sulfa, and chlorphenicol. It not only caused bladder infection, but it caused pyelonephritis and bloodstream infection. And it's distributed all around the world. And so we asked the question, how did this clone distribute itself to all over the world? Is it distributed by food? And we have evidence that UTI may be a foodborne disease. Okay? So our next series of studies went to try to support this new hypothesis. So where did ST69 come from? We know that the enterohemorrhagic E. coli, right, EHEC, is foodborne disease, right? We have an outbreak of this every 2 months in the United States. So why not E. coli that causes UTI? You ingest the E. coli through contaminated food, it colonizes your intestine, and if you're a woman, you colonize your bladder from the E. coli that's in your intestine, and you develop UTI. Okay, so our first suspicion was to look for the source in animals, food animals. So we obtained E. coli isolates from Penn State, which serotypes E. coli from all the animals and humans. And we found ST69 in 11%, 22 from cow, 10 from turkey, 3 from chicken, and 13 from water, and 4 from others. In trimethylene sulfa, ST69 was found only from cow and turkey. And you can see the trend. So the ST69 started appearing among the animal isolates in the 70s. It gradually increased. And the trimethylene sulfa, ST69, appeared in the late 80s and continued. Okay? And when we did the PFGE, which is the most discriminating test, you can see here, the red ones, red lanes, are the animal isolates. Okay? And if you look at number 5 and 4, this is a human isolate. It's about 95% identical. Okay, so it's not quite a smoking gun. Do you understand the term smoking gun when we use that term in epidemiology? When somebody shoots somebody, right? If the person is holding the gun that's smoking, you know that that person is responsible for shooting the gun, right? So if it's 100% identical, then you can say that's a smoking gun, meaning that you have isolate from an animal and isolate from a human being. That's a smoking gun. You don't need to do anything more. Okay? Now, when I said this in Salvador last year, one of the Brazilians said, in Brazil you don't say smoking gun. You say lipsticks on the shirt. Okay. So these are the places where ST69 have been identified, including in Rio. So we did a UTI study in Rio de Janeiro, and we found several cases of UTI with ST69. So it's in Brazil too. So you should look for this in other parts of Brazil. Okay. I think this is the end.